the actual biological mechanisms of gene expression, and that will also explain why an allele is said to be dominant or why one might be said to be recessive. The main thing I want you to take from this is that the interaction between the genotype and the environment produces the phenotype, which is a manifestation of gene product interactions. Okay, so what does it mean? What is a gene product? So we have our gene, it codes for a polypeptide. We would have to transcribe our gene, and then we would have to translate it. And then from that, we get our gene product. And it is exactly what it sounds like. It is a product of our gene. And the reason why we use the word gene product is because we may or may not actually get the functioning protein based on what we have in that gene over there. So we might have our good functioning protein, or that gene might be mutated, and then we might have a loss of function protein, meaning is a polypeptide that does not really do what it's supposed to anymore. So within a given organism, its genes, or within a given cell, its genes are expressed, and that produces the gene product. Now, there are a lot of different factors that come into whether or not a gene is actually expressed, and I guess we can talk about this. But everything within the human body, like all the cells, contain our full set of DNA. So a skin cell has the same amount of DNA as something found within our kidney cell, the same DNA. However, the DNA from that are regulated differently. Different proteins are expressed in our skin cell compared to within the kidney cell. So that allows differentiation to happen. So yeah, and that is because they have different gene products. So for diploid organisms, and that means having two sets of chromosomes for each one. So they have a bunch of homologous chromosomes. So that would be the presence of homologous chromosomes. And this should really be cells, not really organisms. Homologous chromosomes. And then that would be two sets of chromosomes. This could happen twice. Because we have two homologous chromosomes, that means we have two possible alleles because they share the same kind of gene type. So each allele could be expressed and produce their own gene product. So each allele can be transcribed and translated to produce that gene product, and that would happen twice for each organism or for each gene. For a haploid organism, that would just happen one time because they only have one chromosome for each. They only have one chromosome, which means they only have one set of genes possible. Okay, so let's go back to over here. So the interaction between genotype and environment. So there are a lot of different things in play that can actually determine genotype expression. It could be I don't know, nutrition, even altitude. I don't know, just like a bunch of different things that can influence whether or not our genotype is actually expressed. And that produces the phenotype, and then that phenotype is the result of the gene product interactions. And what do I mean by gene product interactions? Well, because diploid organisms can potentially have two competing gene products, they can counteract each other, they can work together, and that would produce a different phenotype. We're going to talk about gene product interactions of diploid organisms. This is stated on the study guide, but it won't be on the exam. So maybe just sit back and chill out, because I think that it is useful to learn conceptually. Cool. So let's say that we have a haploid cell or a haploid organism. So that means that it only has one set of each chromosome. So this is a chromosome. There's a gene there. There's a gene there. There's a gene there. So for each gene, the gene product that it produces will form whatever phenotype is associated with it. But for a diploid, we do have two sets, like humans. We do have two sets of chromosomes, and these are homologous. So that means that we have two genes over here that code for the same kind of thing, but they can be different from one another. So what happens is that you have the potential to get two different gene products, so two different polypeptides most likely, and they can interfere with one another. And that is the interactions we're talking about. So just because you have one thing that codes for a trait doesn't necessarily mean that the trait will actually be observable because of confliction from the other one. So there are three situations that are over here. We're looking at a flower, and we're looking at the color of the petals. So let's say that the genotype for each one is big R, little r for their petal color. So our big R allele is said to be code for red, and the little r is said to produce the phenotype of white petals. Okay, so looking at this example, we see that the petals, even though each one of them are heterozygous, meaning you don't have like two dominants and two recessives, that this one is completely red. We don't see any hint of white in there. So this is a case of classic dominance. What could be a biological explanation for this? Well, to review, with diploid organisms, both alleles can be expressed. They can be transcribed, and then they can be translated by the ribosome, and they both can produce a gene product. But let's say that when the big R allele is red, it produces the red pigment. It's a protein that causes the red coloration. But what happens when the little r allele is red? This one might have been mutated down the line, so it is dysfunctional now. So it might be we just produce a polypeptide, but it is no longer a white pigment. It doesn't really do anything. It's still made, but it's not really like a pigment protein anymore. It's just like there. So what happens is that there is only one kind of protein that can influence the color, and by default, that is just red. Now, let's say that we had the little r, like a homozygous recessive phenotype. We would see that there are only white petals. But why might that be the case when the little r ultimately codes for a dysfunctional protein? It could be that the petals are naturally white without the pigment. So when you don't have anything at all to influence the pigment, it's just white. Now, you can also have a dysfunctional allele where instead of there being a mutation within the coding region such that it results in a different protein, you might not even have the promoter. So remember, the promoter is what it allows transcription to happen. And if you had a mutation that deleted the promoter, then you just wouldn't produce any gene product. So you would still only be producing that red, and then that red is the only one there by default. In this case over here, we have pink, and we know that pink is a blend between red and white. So this is an example of incomplete dominance. We can see that neither one is able to distinctly show the phenotype associated with it. So we still have the genotype big R, little r. The biological mechanism that might explain this would be big R produces the red pigment, a protein red pigment, and the little r produces a white protein pigment. So over here, in this example, I gave a loss of function protein where it didn't even code for a pigment, but this one does code for a pigment. So if you have, by nature of the petals, if you have red and white and they're all spread out, then like the proteins are very small. And then when you look at it from like the naked eye, it's going to look pink, and that would be its phenotype. Neither one of them are dominant. Both of them are there, but like they can't like overcloud each other. And then over here, this is an example of codominance. So the red was dominant over here, or the big R was dominant over here, and we can see distinctly the phenotype it produces. These are codominant because we can distinctly see the phenotypes of both of them. We can see red and white. So this means that, what would be a biological mechanism for this? So like R and R still produces that red pigment, and little r also produces that white pigment. But for whatever reason, they don't really mix, so you can distinctly see the phenotype of each one.